Welcome my students. My name is Musa Shehu. I am here to present a particular topic in biology. Now the topic we are going to discuss is called digestive enzymes. We are going to outline the following. We are going to look at the definition of digestion, definition of enzymes, the types of enzymes, the characteristics of enzymes, as well as the functions of these enzymes. Before we move on for the definition, I will want my listeners or my students to make an assumption. We have been taking in food from time immemorial. Now, have you ever asked yourself that fried rice you have eaten, that jollof rice with that sweet aroma that you have eaten, what happens to that food substance? After some time, you discover that the same food you have eaten through your mouth, it's now going to be removed out of your body through another means and in a different form. Have you ever wondered why that happens? Maybe it is because this has been a tradition from childhood when our brains are not developed to the extent that we can analyze things. So we have been defecating, but assuming someone will be given birth as an adult, and you have just eaten your food, and after some hours, you begin to defecate. You will begin to wonder why. I believe by that, you should be able to ask that question. Why is it that that type of food I am taking in is coming out as a different entity? Now, the simple answer to that question is that food substance we have been taking in, it has to undergo through series of chemical reactions. Now, these chemical reactions, they are controlled by certain organic catalysts. Now, this series of chemical reaction is what is known as digestion. So, digestion in simple terms is simply referring to the process by which complex and large food substances that are usually insoluble are breaking down into simple as well as soluble substances for easy absorption of the body or for easy absorption by our body. Now, these food substances we are taking in on daily basis, our body requires food substances in different forms, and these food substances has to be converted into the form by which our body cells can utilize them. And that is the essence of this digestion. Now, what brought about this digestion is simply some organic catalysts that are called enzymes. Now, what are these enzymes? They are organic catalysts that speed up the rate of chemical reactions in our body. But themselves, they are not affected by the reaction. What does that mean? If we said they are organic catalysts, meaning they are made from natural sources. And a catalyst is any substance that has the ability of altering any chemical reaction. So these enzymes, they have the ability to speed up the rate at which chemical reactions will proceed. Otherwise, so many chemical reactions will have been proceeding at a very slow rate. Now, assuming you have taken in food substance and that food gets digested in 3 hours, that means in the absence of these enzymes, it may take longer time for that food substances to be digested. As a result of that, that is why these enzymes are so important as far as digestion is concerned. And that is why they are called digestive enzymes, because they are primarily concerned about the process of digestion and nothing more. Now, from there, we have so many different types. We have basically 2 types of these enzymes, this digestive enzyme we are discussing. Now, the first one is they are called intracellular enzymes. The second one is they are called extracellular enzymes. Now, what do we mean by intracellular enzymes? These are enzymes that function inside the cell of a living organism, meaning that their function is within the cell of a living organism. An example of these enzymes, that is where we have deoxyribonucleic acid known as the DNA polymerase. You have another example of this intracellular enzyme known as the RNA polymerase, and among others. Now, the other group of enzymes known as extracellular enzymes, these are enzymes that function outside the body of the cell. They function outside the cell, meaning that they can be found, in this case of digestion, they are always found within the alimentary canal. That means from the mouth to the anus. So they can be found within this canal. And that is why they are said to be called extracellular enzyme, because their function is not within a cell, but outside a cell. An example of these enzymes, we have the lipases, we have the amylases, we have the proteases. All these are digestive enzymes, and they belong to this category of enzymes known as extracellular enzymes. Now, these enzymes we have been talking about, they have quite a number of characteristics. They have quite a number of features that they can be identified with, among which is all enzymes are protein in nature, meaning that all enzymes are made up of what? Proteins. That is why they are said to be called enzymes. And all enzymes are made up of proteins. Now, the next we said, enzymes speed up the rate of chemical reactions. What does that mean? Chemical reactions that are taking place in our body cells, ordinarily they will have been proceeding at a very slow rate. But because of the presence of these enzymes, they help to speed up the rate at which these chemical reactions will take place, particularly in the issue of digestion. When you eat food, you are expected to be provided with energy and so many other nutrients that are derived from that food substances. Now, these enzymes, they help in that process so that the whole process is fastened and the availability of energy and other substances of the food are being removed from that food substances. Now, the next we said enzymes are produced in small or minute quantities, meaning that these enzymes, they are always produced in a very little quantity. They are not produced a large quantity. So they are always produced in a very little quantities in a particular place. These enzymes also, they are not used up during the chemical reactions, meaning that at the end of every chemical reaction, these enzymes are not used up in that reaction. They still remain waiting for the next process to proceed. Assuming you have eaten your food and these enzymes partake in the process of digestion, after the process of digestion has been accomplished, now those enzymes are not used up in that process. They will now wait behind and they will continue with the subsequent processes. So enzymes, they are not used up during chemical reactions. Now they act best at optimum temperatures. What does that mean? We said optimum temperatures are temperatures that are suitable for optimal performance of organisms. Now these enzymes, they act best at optimum temperature, and that is the body temperature, which is 37 degrees Celsius. Meaning that an increase in this rate, when you have temperatures far above 37 degrees centigrade, these enzymes will be denatured. They will be destroyed. So they are no longer useful. And when you have temperatures far below 37 degrees Celsius, they become inactive. So they are best when they have temperatures ranging from 37 degrees Celsius to about 38 degrees centigrade, So this is their optimum temperature range. The same enzymes, they are sensitive to pH of their environment. Now, what do we mean by pH? Is the degree of acidity or alkalinity of a particular substance. Now, these enzymes, as I said earlier, they are found within the digestive tract. Now, within the digestive tract, there are various points by which they have certain pH. Now, these enzymes, they are specific in terms of pH. Some of them work best when they are in a pH that has acidity. Others works best in an alkaline pH, whereas there are some enzymes that they work best at a very neutral pH. So the degree of acidity of alkalinity is a factor that these enzymes works on. Now, these enzymes we have mentioned, they do not lose their chemical composition at the end of a reaction. Now, these enzymes, because they are made up of proteins, they are made up of chemical compositions. So, at the end of every reaction, they do not lose their chemical composition. So, they remain the same enzymes, all things being equal. Now, these enzymes, with the characteristics we have just mentioned, they are so important, and as a result, we have the following functions. Now, one of them is that we said enzymes, specifically proteases, they help in breaking down proteins into amino acids. What does that mean? Now, when you take in food that contains proteins, now that protein cannot be utilized by our body cells except it has been converted into its utilizable form. And proteins can be utilized by our body cells when it is in form of amino acids. And it is these enzymes that are responsible for that conversion of proteins into amino acids for utilization by our body cells. And particularly, we said these enzymes that saddle this responsibility, they are called proteases. Now, the next we said enzymes, specifically lipases, they help in breaking down fats and oil into fatty acids and glycerol. Now, when we said fat and oil is the same thing as lipids. Now, fats are the solid part of the lipids, whereas oil is the liquid portion of that lipid. Now, this fat and oil, when they are taken in by an individual, they need to be converted to the form by which the body cells can also utilize them. And that form is in the form of fatty acids and glycerol. So it is these enzymes that are able to carry out this process of conversion of these fat and oils into glycerols and fatty acids. Now, another group of enzymes called amylases, they help in breaking down of starch into glucose, into fructose, as well as galactose. All these are simple sugars. And we said these enzymes called amylases, they help in the process of breaking down this complex starch molecule into simple starch, such as the glucose, which is a simple sugar, the fructose, which is also a simple sugar, the galactose, which is also a simple sugar. And our body cells can be able to utilize all the energy, all the starch in these three forms. These enzymes, they also help in absorption of digested food through the addition of water to the food. Now, after digestion, some amount of water is being added to that food substances so that that food can be digested. What we have to understand in the process of digestion is that not all the food substances we are taking in, that is all being digested. Assuming you have taken in 10 grams of rice, not all the 10 grams of that rice is going to be digested. Part of it will be digested and assimilated by our body cells, whereas the remaining other parts will be removed through the anus. And those ones that are being removed, they are removed in form of what? Feces or fecal matter. And that process that involves the removal of undigested food substances through the anus is known as what? Digestion. Now, why I am still emphasizing on this fact is that you may think you can eat food, after some time you will now vomit that food. That food has not been digested, but you cannot call that as digestion because that is vomited. For digestion to be said, it has to be the removal of undigested food substances through the anus, and that is why those foods are said to be called feces. Now, these enzymes, they speed up the rate of chemical reaction in the body. As we've mentioned earlier, they are responsible in speeding up the rate of that digestion. Otherwise, that process of digestion will proceed at a very slow rate. These enzymes particularly, they bring about reversible reactions. For example, the reaction of starch, conversion of starch to maltose. When we said a reaction is reversible, it means it can go or it can proceed in two ways. That means starch can be converted to maltose through a process known as hydrolysis, that is addition of water. The same maltose can be converted back to starch. And that process is called a reversible reaction, when a substance can be converted from one form and it can be reversed to the previous form. So these enzymes plays vital role in the process of conversion of starch to maltose and conversion of maltose back to starch substances. Now, without these enzymes, almost all the biochemical activities of the cell or of the body will have not make any sense, because so many processes will not be able to take place. As such, so many chemical reactions cannot be possible. And when they cannot take place, it means the possibility of even living will be something to worry about. Now, I have a simple table that summarizes some of these enzymes. Now, this table, as you can see, is a table showing various enzymes, their sources, their locations, the substrate they acted upon, and their effects or the products or their products. Now, number one, we said we have an enzyme called thialin. Now, this enzyme, where is it found? That is the source. You can have this thialin from the salivary gland. That is the saliva. It is the saliva that has the responsibility of producing this enzyme known as thialin. Now, the substrate acted upon. What do we mean by that? It means that these enzymes, because they are specific in nature, they act on certain food substance. Now, if you have eaten a food that contains proteins, contains fats, lipids, and so many other things. Now, the enzymes that are responsible for digestion of starch will always be concerned about those food substances that contains the starch. So these enzymes, they act on starch. And what is the product? When they act on starch, what happens? It results to partial hydrolysis of starch to maltose. It means that they convert starch to a simple sugar known as maltose. The next we said is another enzyme called renin. Where is it found? That is the source. It is found in the gastric gland. Where is the location of this gastric gland in the alimentary canal? It is found in the stomach. Now, which food substance does it act upon? It acts upon proteins. It is not concerned with carbohydrates. It is not concerned with starch. It is not concerned with fat and oil, but it is only concerned with proteins. What happens when it acts on proteins? It coagulates or curdles milk, meaning to change a liquid into solid, into a semi-solid. So that is what this particular enzyme does to proteins, particularly milk. It coagulates the milk, meaning that it converts a liquid milk into a semi-solid milk. We have another enzyme called pepsin, found in the gastric gland, and this gastric gland is also located within the stomach. And it acts upon proteins also. So it acts upon proteins. And what happens after the activity? It converts proteins to peptones. That is another form by which proteins could be converted. That is a much more simpler form of proteins. The next we have the pancreatic lipase. It is, the source of this is from the pancreas. Where is this pancreas located in the body or in the alimentary canal? It is found in the duodenum. That is part of the small intestine. What food substance does it act upon? It is lipids, that is fat and oil. What happens? It converts fat and oil into fatty acid and glycerol. We have another enzyme called amylase. Now this amylase, it is derived from pancreas. It is also found in the duodenum. It acts on starch. What happens? It converts starch into maltose. That is another form of simple sugars. We have trypsin, which is also an enzyme. This enzyme is, the source is the pancreas. It is located in the duodenum. It acts on proteins or peptones. When we said proteins or peptones, these peptones is a simpler form of proteins. So it converts proteins or peptones to polypeptide. That is another form by which the proteins do exist. We have erepsin, another enzyme, very important enzyme. Where is it located from? The source of this erepsin is the sulcus entericus. That is where its source is. Where is it located? It is located in the small intestine. It acts on food substances known as polypeptides. And what happens? It converts polypeptides into amino acids. So if you look at this table, you discover that this trypsin will convert proteins to peptones. And it has done its function. Now, erepsin will now take from that process. When proteins are being converted to peptones, still, that is not the form by which the body can utilize the protein. They still need to be further broken down. And that takes place by the erepsin. So it picks those polypeptides and it converts them into another form, of which is the simplest form of protein, known as the amino acids. So this table have summarized some of the activities of these enzymes, ranging from their sources, their locations, and the substrate, that is the food substance by which they act upon, and the resultant effect, that is the product. Because when they act on certain food substances, there is that conversion from one form to another. So with this table, I think we have summarized what happens to all these food substances. On this note, we've come to the end of today's class. Now I have an assignment for you. Question number 1A, state 4 characteristics of enzymes. Question 1B, Roman 1, list 2 digestive enzymes produced in the duodenum of humans. Question 1B, Roman 2, name the substrate that each enzyme listed in question 1B, Roman 1 acts upon. Question 2, state the product of each enzyme activity in question 1B, Roman 2. Question number 3, in a tabular form, provide the sources, locations, substrate acted upon, and the effects or the products of the following enzymes. 1. Lipase. 2. Lactase. 3. Maltase. 4. Sucrase. On this note, we've come to the end of today's class. In case you have any question or you need more clarification, I can be reached on this phone number 080-33-35-6076. Thank you until we meet again.